speaker, Dr. A. Kadaral Pandian, Senior Scientist, ICAR National Bureau of Fish Genetic Resources, Kochi. Dr. A. Kadaral Pandian is currently working as Senior Scientist at ICAR NBFGR, Kochi. He completed his PhD in Fish Biotechnology from ICAR, Central Institute of Fisheries Education, Mumbai. He is serving as a scientist at ICAR NBFGR for a period of 12 years. He served as an associate professor and head of the Fisheries Biotechnology Division at Institute of Fisheries Postgraduate Studies, Tamil Nadu Dr. J. Jayalalitha Fisheries University, Vanyanjavadi, from 2019 to 2021. He published several research papers in peer-reviewed journals in the field of structural genomics and DNA barcoding. He was a part of in team organizing national and international level training programs in the field of molecular markers in fisheries research. I would like to invite Dr. A. Kadaral Pandian, Senior Scientist, ICAR NBFGR, Kochi, to deliver a lecture on molecular taxonomical methods, tools for resolving ambiguities in taxonomy of aquatic organisms.
Yeah, thanks, Hema, for the introduction. Shall I share my slide actually? Am I audible first of all? Hello?
Yes, sir, you're audible.
Yeah. Shall I share?
You are not audible, sir.
Now? Yeah, just a second.
Volume. So please unmute yourself and increase the volume in speaker.
Hello? Yes. Is it audible now?
So you're audible, but your voice is very slow. Volume is very less.
But I have kept my volume maximum. I don't know. Is it okay now? Hello? Hello?
Sir, yes, sir.
Yeah. Yeah. Hello?
Yes, sir.
Sir, Kadir sir, audible.
Okay. Maybe those who are there virtual, no, for them it is okay. What I feel like. I mean, okay, va?
Yes, sir.
Okay, pa. Maybe there, no, we just check it. Maybe in virtual this thing, no, it is fine.
Okay, audible.
கேக்குதா இப்போ
Yes, sir. Yes, sir, you are audible.
Okay, Hema, thanks for the brief introduction. Shall I share the slide now?
Yes, sir. You can share.
Yeah. Yeah, is it audible now?
Yes, sir.
Shall I start?
Yes, sir.
Yeah, okay, ma. Yeah, thank you very much for the opportunity. I would like to thank the organizers, especially Dr. Raghuran sir, Dean of my alma mater, where I have studied my UG, the batch of 95 to 2000, long back. This is, and good morning to all, those who are participating in this particular workshop. In a nutshell, I would like to tell about what is the kind of utility of molecular taxonomy. As you people know, as Jare Raja was mentioning, like some UG students also there, they may be learning about the taxonomical tools in fish identification. I would like to add up on some kind of application of molecular taxonomy in combination with the traditional methods, how effectively we can use it for species delineation purpose. Yeah, this is our institute. I will start normally with the institution kind of brief introduction. This is the National Bureau of Fish Genetic Resources, as most of us are aware, and mainly we work on conservation of fish genetic resources of the country, and not only that, and we'll be striving for utilizing that bioresources for the prosperity as well as for the livelihood development. Yeah, this very topic is what, like how we can use this molecular tools in species identification in effective manner. It is not like everywhere we'll be using this molecular taxonomy. In some of the circumstances wherein this traditional taxonomy may not give provide information, in those cases we can able to use this molecular tool in an effective manner. As you know, like these are the kind of thing with using this molecular taxonomy, which is normally called as DNA barcoding. We can able to identify at any life stage. Actually, if it's a bigger one, it is not much kind of issue. But having said that, some kind of ambiguities also we need to use this tool. And the other processed product and even like ichthyoplanktron, like egg and larvae, we can use this thing in an effective way. As you know, like no, that is a full organism or full specimen, whatever I was talking about. Here, when it is come to the meat, actually here you can able to see these two things are like often do different species, but when we see this particular chunk of meat, we may not be able to like identify using this very traditional kind of taxonomical tools, wherein we can use this only. I'm underlining this molecular tool only can be effectively utilized for identifying this kind of meat which belongs to which species, right? Again, when it's a processed product, we won't be having those attributes which are helpful for identification of the species or whatever. But here you can see, like once it is a product, processed product, I say it's a canned one. Like we cannot differentiate the group. Like actually, this may be like from different species, and this may be other species of the same genus. But these cases we can use this molecular tools in an effective manner. Yeah, this is a nutshell, like how we can utilize these molecular tools for our taxonomic identification.
Excuse me, sir.
If it's individual, yeah, hello?
Sir, slides are not
Yeah.
moving. You can keep it in your PPT.
Yeah, it is already in PPT view only.
Okay, I think slides are
It's
not.
like,
Slide. I think slide is in first slide.
yeah, this is a title slide,
Yes, sir.
right? Just now, whether is it okay? I have changed it. 4 slides
No, it's not moving.
so
Sir.
far.
Slides are not moving.
Okay.
I think you can share the entire window, sir.
Okay.
Yes, sir. Now it's moving. Yes.
But this is not in slides, so but is it okay?
Yes, sir. Yes, sir.
Is it okay? Okay, that's fine. Something is, yeah, again, slide, I will try like once again, slide mode. Is it okay now? No. Not moving, right?
Yes, sir. Not moving.
Okay. This is fine, right?
Yes, sir. Now it is moving.
Okay. Yeah, thank you. Thank you, Susan. Actually, again, I come back, the things like whatever has been explained so far, like in adult specimens, even different life stages, we can able to identify the kind of species in a very effective manner using only traditional molecular tools, wherein when it comes for the meat kind of thing, like what we can say, like these are the two different species, but when we make it a chunk kind of thing, that we cannot able to differentiate this both the things of belongs to which species. In this case, we can use this molecular tools in effective manner. Similarly, like when it is a processed products, we may not be able to identify the species origin using these traditional tools, wherein we can use this DNA barcoding as an effective manner, right? In a nutshell, like we can say, like how we can use these molecular tools for species identification in different scenarios. Wherein first we take like individual species, we can use both the things. In certain cases, like cryptic speciation, for example, what is cryptic speciation? You may be studying for, especially for UG students. Actually, we presume like the particular area we have only 4 or 5 species of that particular genus or whatsoever. But when we do in-depth study, that time we could able to come up with more number of species. In this case, traditional taxonomy, it is a kind of use, but it is molecular tool can able to delineate the species whatever present in the particular system. Again, some deformed sample when we go for sampling for our research, and sometimes we will be getting the specimen, maybe not many numbers, 1 or 2, that also in a deformed condition due to the netting activity and whatsoever. In those cases, traditional taxonomy, we cannot use it effectively. We can use the DNA barcoding easily, because for a DNA barcoding, we need only a small portion of any material, like starting from meat, like muscle or even fin or whatever it is available in a very, very, very minute quantity, we can able to identify the particular species origin. Again, as I was mentioning, like processed meat, again, it is a kind of issue with traditional methods wherein DNA barcoding we can use it. Like ichthyoplankton, I was telling, like even some kind of again larvae, but some cases we can able to identify. In most of the cases, even molecular taxonomy using this DNA barcoding also, it is much more difficult because it is a mixed kind of thing. Like only DNA barcoding we can effectively use where it is a individual kind of samples are available. Some other methods are there that we can able to do in advanced DNA barcoding using like next generation sequencing metabarcoding, we can say, in that a group of organism in a particular sample also could be effectively identified. See, this particular concept of DNA barcoding, it evolved from the product barcodes, right? Product barcode in the sense when we go to the supermarket or even a small kind of stationery shops or even grocery shops, nowadays the people, they have the barcode. They scan the barcode so that the entire information is available in the barcode, right? We can able to identify what kind of product it is and what kind of other information it contains, including the price and other things. Similarly, like this was conceptualized like as you know, in DNA, that is a nucleic acid, that is the main kind of a constituent unit of function of particular organism. In that, we know like it consists of 4 different nucleotides and different combination. By studying this kind of arrangement, we develop some kind of barcode kind of information, like what is it is available for the product barcode. That is a concept like the arrangement of these 4 different nucleotides in a particular area in the genome. Not the entire genome, we cannot study for these kind of purposes. Lot of studies were put in this finding the particular gene of interest that could be able to differentiate between the species. Yeah, this is called as DNA barcoding pipeline. Pipeline in the sense how the workflow flows through. But initially we have to go for a specimen collection from wherever it is intended. And again,
Excuse me.
Yeah.
Can you make it as a slideshow, sure?
But slideshow it is not moving, ma'am.
Okay, sir.
I tried actually, but it was in slideshow initially, but it was not scrolling down.
Okay, sir.
Is it, Mama, only a few slides only, it will be like small actually. Other than that, I feel like it will be okay.
Yes, sir.
Yeah. This is a tissue we'll be taking out of the specimen, whatever we have collected, and we will be isolating the genomic DNA. That is called DNA processing. And we will be like amplifying PCR. Most of us know, like particular gene in that entire genomic DNA, we will be amplifying. For example, 650 base pair product of mitochondrial origin, that is called as cytochrome oxidase 1, is commonly used to differentiate all the heterozoans. Here, once the PCR is done, then we can do the sequencing, And once it is done, by using the different statistical tools to analyze the kind of information it has been generated through DNA barcoding, and so that we can have inference on the origin of the particular sample which belongs to a which species or whatsoever. Here, some different tools are like databases are like there. I will explain later. But this is the kind of concept what we are using it. Once it is done, once the barcode is the sequence information is generated, it will be like published in the public domain, so that those who are using in the studying later in the particular group, they can effectively utilize this molecular information for their studies also. Yeah, that's what I was mentioning, like CYA gene, it is generally used for DNA barcoding. Here, why it has been selected, like it was tested like several genes in the genome. They were tested for this particular concept or particular utility. At last, that cytochrome actually is one of mitochondrial origin. It is not a nuclear gene, it is a mitochondrial gene. We have different kind of advantages by using the mitochondrial gene, because it will be having more in copy number, and also they are not under the selection pressure that is normally happens in the nuclear genome. And also the main thing is that that is a maternal origin. Mitochondria, as you know, like they are maternally inherited, so that for evolutionary studies or even demographic pattern analysis, we can effectively utilize this particular gene. Not only this gene, different sets of genes of mitochondria, they are being used, because here you can say like we can use a universal primer, the sense single set of primer. We can use it for a species across all the kind of order or whatsoever, and also it can be able to differentiate even closely related species also. Yeah, these are the genes. Generally, we use it like metageomes, cytochrome oxidase 1 gene, and again we use like 16S rRNA, ribosomal RNA, and another gene like cytochrome B also, it is being commonly used in metazoans. When it comes to fungi, it is ITS 1 and 2, they are nuclear gene. They are used for DNA barcoding. Similarly, like algae, when you are working in algae or any plant-based kind of samples, we'll be going for Maturus K and RBCL. They are like metazoans, it is a kind of mitochondrial origin. Here we are going with chloroplast, which is almost like similar to the mitochondrial origin in animal species. Then again, some mollusks, a lot of the genes like even COA it is used, and 6S and 12S rRNA, these are mitochondrial origin in histone 3. That is also another gene that is commonly used for mollusk, which is from nuclear origin. Yeah, once we have developed the information, we can submit into the NCBI, National Center for Biotechnology Information, where we can get the assistant number, because that is very mandatory for publication of our findings in reputed journals. There is another thing, this is another kind of database which is available in the public domain. This is called as Barcode of Life database. Here also like assistant number, what is being provided by the NCBI, here the bin numbers are provided, and also it is a kind of another utility wherein we can have some kind of tutorials to learn these barcodes, whatever it is happening. That will be very much useful for the students, and also like those who are working in a same kind of organism across the globe. Here the provision of workbench also there, so that they can able to share their knowledge and they get the like clarification if anything required. Yeah, this is a kind of nutshell what I have explained about the barcoding process and what kind of genes are used and whatsoever, and also like application, like that's a general way it has been discussed. But I want to take you to some kind of specific kind of examples, like in our lab and whatever we have done so far in using this DNA barcoding. That will give much more information in understanding the utility of this DNA barcoding or molecular tools for species identification. Just a second. Yeah, first of all, we initially, we were very much interested because NBPR is an institute which started this DNA barcoding first from not only India, Southeast Asian country, like in 2005. In those days, in 2007, we have received a sample from Forest Department of Kannur, Kerala. Therein, this, as you know, like scheduled species, what are the species which are categorized under Schedule 1 of Wildlife Protection Act? They are banned for capture, but what the fishermen, they do, like they capture these organisms and they make into a small kind of pieces, meat pieces, and even fins or whatsoever they will bring into, for in case of shark fins, they will be cut and they bring to the landing centers. But forest officials, they know like this may be the protected species, but still they have to prove the kind of identity in the court of law. In that case, the samples were sent to us, and we could able to develop this cytochrome oxidase 1 and other supportive genes, and we could confirm that specimens which were confiscated are of whale shark. That is a protected species. Why I mentioned this particular thing is like the molecular evidence, whatever it has been produced by us, that is accepted in the court of law. Okay, that is the kind of utility of this DNA barcoding. This is the one case we have come across in 2007-8. Even this was like much more recent, that to our Gulf of Mannar. Here also you can see how you might be knowing the mammal is protected under Wildlife Protection Act. They are not supposed to be caught. And similar kind of issue has been came to notice to the forest officials, and they sent the meat, whatever it was confiscated from the fisherman. And by using this DNA barcoding method, we could able to identify this origin of this meat, and that has been produced in the court as evidence. Yeah, this is another kind of interesting application of forensics, DNA barcoding in forensics. This is a kind of different kind of scenario, like what I mentioned in the previous two slides. Here in Mumbai, one peculiar kind of case, like it has come to the police knowledge. Actually, what happened? One lady, she had gone to a restaurant, 5-star restaurant, and she ordered for this farm prep for curry, but they served the particular fish, but she was in the contention like that particular dish was served was not of farm bread. Then the complaint raised to manager, and that way it has gone to even police, and they seized the sample, and they have sent to us for confirming the identity. That is a product actually in curry as well as fry condition. We developed in our lab for the methodology for even fried kind of fish items also to generate the molecular sequencing identity. That we could able to identify the samples like they are farm bred only, not Fambus argenteus, that is the most sought out. That is a Fambus sinensis, some other species, but what it is farm bred only. That was a kind of findings we could able to give it to the court of law. Then again, next when we go to some other areas, there is a forensics. I mentioned what are the application of this forensic application that is very much needed for this our when we are going for strict implementation of fishery management practices. Next, it is when we study, even if you study taxonomy, you might be studying about the finfish, maybe shellfish taxonomy. I hope that some separate course is there in recent times. But when we go for this particular kind of molluscan species, they are called as Conus species. We had a project from DBT for this particular thing because nobody has done this molecular kind of thing in Indian waters. Here you can able to see here these two things. Mostly these cones, very selected features like spire length or whatever 3-4 morphomeristic characters, and main kind of identification attribute is coloration only. But sometimes we collected these kind of different colored specimens, and when we checked this kind of morphology also, it was totally matching. Then we got intrigued, like whether because earlier there was no molecular information, it was available from Indian water. We have standardized that thing for 4 genes, whatever mentioned here. But when we checked with the molecular information, we could able to come up with the conclusion like these two are of same species but different morphotype. Here also you can see these are 3 species we have given as a kind of example. Here, this particular case also we can effectively use this molecular tools. Otherwise, we might have thought like these are of different species. That goes to the wrong kind of conclusion of our diversity studies. Again, here picture, you ignore the picture because some of the larval stages are missing here. Again, for larval stages in Lampis, this is another Strom of molluscan group. Here, like considerable morphology difference is there, but still some of the stages we thought like that is of different species, not from this particular Lampis. Then we collected and we could able to like get the information through DNA barcoding that what are the young ones are belonging to this Lampis or any other species of molluscan origin, right? Yeah, this is another kind of thing. This is next application of DNA barcoding. Actually, when you are describing something new, as like you might have like come across like different species. The species are added to the science every time, maybe every day something or other thing. It will be keep an order for our kind of studies. Here you can see like across the different kind of group. This is the kind of thing what we have reported and we named against this ICAR, our organization, Pankaj Icaria. It is from Metur Dam. It has been described last year. And again, even from Conus, we could able to describe here new species from Lakshadweep water. This is another yield of marine kind of thing. We have kept on diversity kind of things like any kind of group of organism. This particular tool of molecular taxonomy could be effectively utilized across the different group of fishery or even any organisms, like not only like fish or whatsoever, and other groups also. Whenever you study in different kind of thing, then there also we can effectively utilize this molecular tools for species identification and new species discovery also. Yeah, this is the species what we have got, Conus species from Kalpeni Island in the Lakshadweep. You can see like this way, this is the original kind of picture taken when we first identified this. It means where first we noticed this particular species specimen. But this is the kind of polished one after when we brought to the lab, and this shows like where it has been like collected this new species. This is new species. Yeah, this is the kind of molecular information. Maybe it is much more smaller to see like, but just to have an idea like how we are using it, like based on the molecular information generated, we compare with the closely related species, whatever it is available with the National Center for Biotechnology Information, and we generate some kind of module. Here, this is kind of Bayesian inference. Normally we go for different modules like neighbor joining or whatever. This is like maximum livelihood model. We generate the phylogenetic tree. Phylogenetic tree, it denotes like the closeness, the evolutionary status of different species in a particular study. You can able to see here, these 4 or 5 are new species sequences. It is delineating clearly with the closer one. Closer one is Conus distance, but here we could able to get the clear-cut kind of topology coming out of this particular group. We can infer like this is of the specimens what we have collected and confirmed with morphological in some cases, then we can fully confirm, because when we are sending for publication, this is a very much essential in recent times. These are some sequences what we have used for the study. This is a basic kind of inference compared to this module. Like here, we will be getting more kind of information. Like this has been used for our Pankaj's Icaria in the recent publication. Yeah, again, some of the other kind of adaptation studies also we can able to information about the species adaptation also we can infer through this particular molecular markers. What we have done it like it is not an intentional kind of study. What we have collected samples from different location in India as well as for island ecosystem of Andaman Nicobar as well as Lakshadweep Islands. But when we are analyzing the data, that time we came to a kind of different conclusion. See here, both the islands, like as you know, like Andaman and Lakshadweep are both are in different location with a lot of distance. There won't be any genetic mix-up possibility. That because these mollusks are not very migratory kind of thing, then genetic gene flow will be much more on the lower side. In that case, we could collect the specimen of same species from Andaman as well as Lakshadweep also. In Andaman also two different kind of system, ecosystem, like one is rocky and other one was sandy kind of system. Similar way in Lakshadweep also we collected. We perceived like maybe this rocky and sandy of Andaman origin and Lakshadweep origin also they were supposed to be separate, like because Andaman it is much more nearer the two ecosystem where we are collected. Similarly, Lakshadweep also we consider like that. That is a normal kind of hypothesis. But when we analyzed the data, it has come to a conclusion like, see that rocky showed collected specimens in the Lakshadweep and same in Andaman. They are much more closer in terms of evolution. And also like for those who collected from other coral areas, they are of same kind of sharing the same kind of evolutionary kind of thing. Here, rocky and sandy of same kind of islands, they are distantly related. This kind of adaptation also we can able to identify using this molecular information. Again, for evolutionary study, what are the species are closely related and what are the species are distantly related in here. How we evolved actually, what is the evolution time of a particular species. When you say like scambrid, this is a recently published paper of the scambrids of India, around 16 species we could able to collect, and we wanted to check it like how they are evolved, like which species evolved first and what the species they could able to the evolution rate of means the period of evolution of a different species also we can able to identify using this molecular tools. Yeah, another small example, like even when we talk about the new record of this particular species, whatever we have collected, nowadays mostly we are using this kind of DNA barcoding for identification of, for confirmation of new records also. See, this I was telling like in scambrids, when we have done this DNA barcoding, some of the groups like in the Thunnus genus, we are not able to differentiate using this cytogram oxidase 1 gene. Then we developed the character-based identification system. In the barcode itself, it consists of 650 base pairs. In that location-specific, nucleotide-specific, any difference is there. Using this character-based identification system, then we could able to come up with the conclusion like for this the group like Thunnus group is which is not possible to delineate using this commonly used cytogram oxidase 1. The character-based identification system would be able to differentiate. This will be helpful in developing the DNA chip for the particular group for forensic as well as fishery management purposes. So these are some character-based identification keys for we have developed for different groups. This is actually for Pulicat Lake. It has been done actually for all the species, but this is for Elasmos branch only. All the Elasmos whatever it is available in India, and actually against scambrids also like it has been done CVAS. Yeah, you shall go to the kind of DNA chip for a particular kind of organism. We have done a project in it for scambrids as well as. Even tuna also, like we could able to come up with some kind of findings, then CBIS based kind of tools which can be effectively utilized for developing the DNA chip for a particular group of organism. Yeah, initially I was telling like when there are mixed, there is possibility of mixed samples as well as mixed kind of product also, like as a tell like even if we are sampling eggs and larvae, we cannot have a single kind of species in it. Maybe multiple species it will be present. In those cases, the DNA barcoding it is much more difficult to differentiate. In those cases, a recent advancement, recent lot maybe it developed maybe before last 5 years, but we are using it recent time. There is a meta barcoding. Here you can see like here each species only could be identified using our normal DNA barcoding method. You take the specimen, you do DNA extraction and PCR, and by analyzing the sequences, and you can get the species identity. But when we have the mixed kind of thing, this particular DNA barcoding, it cannot be used like. But meta barcoding, the concept using the next generation sequencing, we can able to get entire information like what are the species are available in a particular sample what we have. That is a kind of environmental DNA analysis we take from water or even for sediment in the water body. Then we can, without like going for actual sampling. This is called non-invasive sampling from water. We presume it is like, not presume, like it is a kind of studies are there. Even the water that genomic DNA, like it persists like for a particular period of time. Even sediments is much more compared to the water samples. Here you can see like using these environmental DNA samples at recent time, a lot of studies it has been taken up for especially in fresh water, and also most of the studies it has been done for species only. It is a kind of different kind of utility. It could be effectively utilized for our fishery management purpose, especially for exotic species management. Because before getting established into a system, we can able to identify the presence of the particular species in the water body. This is a study what has been done from university only. Like when I was there for a short stint for 2 years, one of my PhD students, they has done this study eDNA based analysis. We selected this Pulicat region, different like collection points, and these are the things. Initially, we checked in vitro, like we have kept these specimens, even suspicious of marine origin, even mullet and whatsoever in a system. We first standardize this technology. This is the thing in vitro, what it has been done for eDNA based analysis. And after that, we collected specimens from like in vivo, in the sense we went to the field and we collected the water samples, and then same kind of methodology, whatever it has been standardized previously in vitro, we could able to get some kind of preliminary information like the that utility of this DNA. This is the primer we could able to develop. And this is the kind of identification at in vitro we could able to get. These are the species with whatever number. Here you can see like this is a 2069 means this many copy numbers are there. Here not only the species identity and also like abundance also we get able to get the information. Here Google supplies in a particular sampling location we could able to get more kind of thing. Maybe egg or larvae or even adult specimen, but total abundance also we could able to get through this DNA based analysis. Here these are the kind of comparison in vivo in the field study, like eDNA and landing center data also be collected. What is the proportion of species available in the Pulicat lake using this eDNA analysis? This is a kind of initial study wherein we get able to come up with the utility of this environmental DNA based barcoding analysis. Yeah, thank you very much for the opportunity and the patience listening. If you have any kind of query, you can able to get it clarified right now. Thank you very much.
Please ask at least one question.
Thank you, sir, for sharing your erudite knowledge on the use of molecular taxonomical tools in fisheries research.
Yeah.
Now, the session is open for discussion.
Make it must please ask at least one question that can be asked next.
Sir, there is a question in chat box from
Okay.
Dr. S. Saravanan.
Yes?
How,
Yeah.
how can we identify both genus and species level identification in terms of morphological and molecular characterization of seaweeds?
Yeah, that's what I was telling. Like in seaweeds, for morphology, some attributes are already described. Using the descriptors, they can go for morphology identification. Wherein for seaweed, I mentioned like cytogram oxidase 1 cannot be used. In that case, we'll be using like MADK or RBCL. That is the genes, no? They are similar to cytogram oxidase 1. We can use that for differentiating closely related species also. There won't be any problem. Yes. Is it clear?
Sir, yeah, very good afternoon, sir. I have one question.
Okay.
So actually, we used to collect eggs and larvae from
Okay.
the ecosystem, and we feel very difficult to identify their means based on the morphological characters. How
Yeah.
it could be better to use it with this molecular technology, sir?
Yeah, that is what I was. Yeah, please. Actually, egg and larvae, when we collecting it is very difficult to differentiate at the species level. But at least you can have some kind of already checklists are there, even field level identification kind of analysis. But you can try to identify at least a kind of group wise, different groups. Then we can go for normal DNA barcoding for individual means collection of 3-4 eggs or larvae. We can do it because we are doing it for some other group outside also, quite kind of thing we have done it. But as I said, like eDNA based kind of thing right now, meta-barcoding, it is called meta-barcoding. Meta-barcoding, the entire samples can be put together and we will go for next generation sequencing, and we can come up with a clear-cut kind of information about the composition of the species present in the particular sampling kind of environment.
Thank you, sir. Thank you, sir.
Thank you.
So we have another question. Can we use cytochrome B gene instead of CO1 to describe a new species?
Yes, we can use cytogram B, but as a normal case, like when cytogram oxidase is considered as a gold standard, it is better to go for cytogram oxidase gene as a primary kind of gene, then supported with cytogram B and 6' rRNA. But with cytogram B only, if you go like, even when you are publishing it, that reviewer may ask like, why you have not done COI? That kind of question will arise that time. It is better to go with cytogram oxidase 1, supported with cytogram B and 6' rRNA. Yes.
Thank you, sir.
Yes. Yeah, ma'am.
If there are no doubts, we will move on to the next session.
Yeah, thank you very much for